The food that we eat goes to the stomach, and from there it is broken down into three things. Specifically, we are talking about the complex sugar that we just looked at, which included maltose, lactose, and sucrose. They are broken down into simple sugars such as glucose, fructose, and galactose, and these are absorbed in the small intestine, and from there they go to the hepatic portal system, which basically transfers the simple sugars from the GI system to the liver. Glucose specifically enters into the liver cells, the hepatocytes, using the with the help of the GLUT2 receptor. GLUT2 is going to basically carry the glucose, and it's going to bring it in via the facilitated diffusion. Therefore, no ATP is needed. And know that the glucose which is inside the liver is stored as glycogen when it is in excess amounts, and glycogen is broken down into glucose again when there is an decreased amount of glucose circulating in the blood. We will cover the mechanisms later, but just note that the glucose is stored as glycogen inside the liver, and also note that the glucose also exists. It exits the liver and goes to the bloodstream for the beta cells or the alpha cells of the pancreas to sense the amount of glucose present. When there is an increased amount of blood glucose present, the GLUT2 receptor, note it is the same receptor which was present in the liver cells, the same GLUT2 receptors from the pancreatic cells bring the excess glucose into the cell, and that excess glucose produces ATP, which ultimately causes the release of the vesicles of the beta cells in the pancreas, release of the vesicles which is inside the beta cells of the pancreas, and these vesicles contain insulin, and insulin, which we have seen, insulin decreases the amount of blood glucose by using glucose by using insulin dependent glucose transport mechanism, which we will also cover in the later lectures. Let us now focus on the specific mechanisms that causes beta cells of the pancreas to release insulin. When there's an increased blood glucose, the glucose that circulates gets attached to the GLUT2 receptor, and it enters the beta cells of the pancreas via facilitated diffusion. And as mentioned, during facilitated diffusion, there's no ATP. Basically, the glucose is going down the concentration gradient, so no energy is needed. Now, once the glucose is inside the cell, glucokinase, which is inside the beta cells of the pancreas, phosphorylates the glucose. Therefore, we have glucose 6 phosphate. And glucose 6 phosphate cannot leave the cell because the negative charge on the phosphate group causes the entire glucose 6 phosphate to be more negative. And note that the resting membrane potential of the cell is negative 70 millivolts. Because of that, the negative charge on the glucose 6 phosphate and the negative resting membrane potential causes glucose 6 phosphate to stay inside because negatives and negatives repel each other. Now, the glucose 6 phosphate, when it is inside the cell, it undergoes glycolysis, um, and in the end it produces pyruvate, and pyruvate goes to the mitochondria and it produces ATP. Now note that the resting membrane potential is actually maintained because of the potassium channel. When the potassium channel is opened normally and the potassium leaves the cell, that's why we have negative 70 millivolts. And when more ATP is produced via glycolysis, citric acid cycle, and electron transport chain, the ATP actually attaches to the potassium channel and becomes ADP there, and it actually closes the channel. It prevents it blocks the potassium from going out of the cell. Therefore, the cell becomes more positive and the cell is depolarized. And when around negative 50 millivolt is reached, threshold occurs and the voltage gated sodium channels open, and it causes more depolarization to occur because the sodium enters into the cell and the cell becomes more positive. When this occurs, eventually the calcium channels open. The calcium channels are also voltage gated calcium channels, and when they open, the calcium enters into the cell and the calcium attaches to the vesicle. Now note that inside the vesicle we have insulin and we also have amylin, proinsulin, and C peptide. Now the calcium is going to attach to the vesicle, and note the charge of the calcium is plus 2. When more calcium binds to the vesicle becomes more positive. And note that the membrane potential, even though it is depolarized, is still negative. So the more positive vesicles is attracted to the negative uh, cell membrane of the beta cells, and it goes there, and it actually fuses with the membrane. The vesicle fuses with the membrane, and we have the release of insulin, C-peptide, proinsulin, and amylin via exocytosis. And just note that proinsulin goes to the Golgi apparatus where it is broken down into insulin and C-peptide. We are not too worried about proinsulin for now because the vesicle contains only a small portion of proinsulin. And just to recap, the function of amylin is to basically delay gastric emptying, so the person does not eat more. And as we already discussed, when there's less amylin, the person gets hungry often and he eats more, and it causes an increase in blood glucose, which when left untreated, yes, the becomes a cause for diabetes. In the next lecture, we are going to consider some clinical factors, and we are also going to see the other places where GLUT2 receptors are located. We are going to use mnemonics to remember the clinical factors, and also the location of GLUT2 receptors.